Good day to all audience watching this video. I am Tan Su-An with metric number 180773, a final year student in Bachelor of Chemical Engineering from Universiti Putra, Malaysia. Today, I'm going to present on my final year project titled Bioremedation of Chromium-3, Chromium-6 and Dye-Chromium complex using probiotics. To understand better, first I would like to introduce my research study. In particular tannery wastewater from textile industry, there's often coexistence of chromium and dye as a pollutant in the wastewater. Bioremedation is one of the methods to control and degrade waste. It is eco-friendly, harmless towards the environment, and more cost-effective way compared to physical method and chemical method. The use of microorganisms, such as probiotics, may have the ability to eliminate toxic minerals from the environment when provided alone or in combination. In this study, the problem associated with toxic metal found in wastewater was due to its widespread of industry use, which have contributed to high concentration of chromium and dye pollutant in a waste and water system which is above the existing control level. Next is the problem associated with the use of microbes to treat the wastewater. It's because it might be time-consuming process to grow the microorganism. Some microbes may be pathogenic. And lastly, it's difficult to monitor the growth of microbes. Next, we are going to talk about the objective of this study. There is two objectives in this study, which are: the main objective is to screen the best microbial strain for degradation of chromium ions and chromium dye complex compound. The second objective is to optimize the degradation kinetic of selected microbial strain in degrading chromium ions and chromium dye complex. In this slide, it's talking about the methodology of this whole study. It was divided into 4 parts, which are the preparation of nutrient broth and pollutants. Second is the growth of bacteria. The third is the microbial treatment. And the fourth is testing of microbial treatment. In the first part, we are preparing the nutrient broth for microbial growth, the isolation of probiotics, and also the preparation of synthetic solution. The second part is the bacteria preparation and cultivation, and also the growth rate of microbial. The fourth is the parameters that we have fixed to run is in different parameters, which are different pH, incubation speed, and the initial concentration of the pollutant. And lastly is to test the degradation and decolorization efficiency. Probiotics of Bacillus subtilis was isolated from natto powder, whereas Lactobacillus acidophilus and Bifidobacterium were isolated from commercial ECO yogurt. Next, we will move on to the results and discussion. In this first part, we will talk about the removal efficiency. In this part, we are talking about the comparison between the monoculture and the mixed consortium between chromium-3 ions and chromium-6 ions. In the red bar chart, it represents the mixed consortium, which consists of Bacillus subtilis, Lactobacillus acidophilus, and Bifidobacterium. For the blue chart, it represents the monoculture, which is Bacillus subtilis. Now, we can conclude that the mixed consortium consisting of Bacillus subtilis, Lactobacillus acidophilus and Bifidobacterium are more efficient in removing chromium-3 ions and chromium-6 ions. Next is a comparison between monoculture and mixed consortium of Dye-Chromium-3 ions complex. In this figure, we can see that there is no difference in monoculture and mixed consortium for dye removal, as both are able to remove the dye efficiently. Whereas in chromium-3 ions, the removal of pollutant chromium-3 ions are more efficient in mixed consortium compared to monoculture. The next one is Dye-Chromium-6 complex. It shows the same trend as the previous slide, where the dye, there is no difference in monoculture and mixed culture, and that its mixed consortium shows a better efficiency compared to monoculture. The next one is about the kinetic model analysis. As you can see here, the red line represents the chromium-6 ions in chromium-6 oxide, whereas the blue line represents chromium-3 ions in chromium-3 nitrate. The linearity of Lorentz concentration versus time shows the first order kinetic reaction. The K value for chromium-3 ions is 0.0514 per hour and 0.0428 per hour respectively. This shows that the removal of more toxic chromium-6 ions is faster than the chromium-3 ions. Next is the kinetic model study for Dye-Chromium-3 ions complex and Dye-Chromium-6 ions complex. The purple lines represents chromium-6 in potassium dichromate. The red line represents chromium-3 in potassium chromate. The blue line represents the trypan blue in potassium chromate, and the green line represents the trypan blue in potassium dichromate. Dye and chromium removal occurred at different rates, judging from the K values. Now let's talk about the dye. Trypan blue dye removal from Dye-Chromium-6 complex pollutant was twice faster than the Dye-Chromium-3 complex, where the K value is 0.2497 per hour and 0.1623 per hour respectively. Interestingly, the degradation of chromium-3 ions and chromium-6 ions occurred at the same rate with the same K values of approximately 0.04 per hour. As a conclusion for this research study, the first objective to screen the best microbial strain for degradation of chromium ions and chromium dye complex compound under various parameters such as initial concentration of the pollutant, mixing speed of incubator, and the pH was achieved. And it was found that the optimum removal is found at pH 7, mixing speed of 100 rpm, temperature of incubator at 37 degrees Celsius with 20 ppm pollutant dosage. Next is mixed consortium gave a better pollutant removal compared to monoculture. The second objective is to optimize the degradation kinetic of selected microbial in degrading chromium ions and chromium dye complex respectively. It was found that the degradation kinetic for chromium-3 ions, chromium-6 ions, and Dye-Chromium complex fits to the first order kinetic model reaction. Recommendation for the future research is to use different range of microbial group such as algae, fungi, yeast, and bacteria to treat the waste containing heavy metal and textile dye effluent. The second recommendation would be to conduct this research in a bioreactor system to simulate the actual industrial application. And lastly is to use the actual waste from industrial to further this research. Thank you so much for your attention throughout this presentation video. I hope this can help anyone out there to understand better. Thank you.